Hey, my name is Macy. I'm going to be talking about the COL1A1 protein. This protein is located within the extracellular matrix of cells. Some examples of these cells are fibroblasts, osteoblasts, chondrocytes, myofibroblasts, and epithelial cells. Synthesis first starts in the rough ER. From the rough ER, with the help of the Golgi apparatus, it's moved out and stored in vesicles, and then vesicles secrete the protein into the extracellular matrix where the protein lives. One disease or condition that is associated with COL1A1 is Caffey's disease. Caffey's disease causes abnormal bone growth in infants in their long bones, ribs, and jaw. It can also cause swelling, inflammation, edema, and hypertrophic bone development. So when COL1A1 is functioning normally, it instructs cells to make type 1 collagen. This is super important because type 1 collagen is one of the most important types of collagen in our body. It helps to support and strengthen our bones, cartilages, tendons, skin, and some parts of our eye. So you can obviously imagine how if this collagen wasn't made correctly, we would have lots of issues with lots of different parts of our bodies. Now I'm going to talk a little bit about the mutated protein. So I'm going to be specifically talking about the COL1A1 R836C mutation. And in this mutation, we have, so first I'm going to talk about a few of the amino acids that are involved in the synthesis. So here we have the primary structure of our protein, and the amino acids that we have that are involved in the DNA of this protein are going to be proline, hydroxyproline, glycine, and arginine in the normal functioning. When the mutation happens, so this is not going to show the mutation, but when the mutation happens, let's say the orange one, the orange bead is our arginine. So in the specific mutation, an arginine is going to be substituted for a cysteine. Now, when this happens, the structure and stability of type 1 collagen is going to be negatively affected. This mutation is just going to make the triple helix structure of the normal protein less stable because of the change in bonds. So essentially, this mutation causes the normal protein instructions for instructing type 1 collagen to not work as well, and it makes less functional type 1 collagen, not less functional, more of less stable, less strong type 1 collagen. So as I mentioned, this mutation can cause diseases like Caffey's disease or osteogenesis imperfecta. Now at the cellular level, I'm going to talk a little bit about what happens at the cellular level in this mutation. So cells' ability to create type 1 collagen is impaired, and so then you have cells that secrete poorly formed type 1 collagen. Now this poorly formed type 1 collagen weakens the extracellular matrix of cells. As we talked about, that's where the protein is located, so it's going to affect the extracellular matrix and make it weaker. Now, this is also going to affect your tissue integrity, and we can see how this is related to bones that are more brittle or weaker tendons, cartilages, etc. So now I'm going to talk a little bit about I think I've already talked a little bit about this, but I'm going to talk about, this is the primary structure of our protein. I'm going to talk about the normal amino acids sequence and the normal amino acids that are involved in our protein. So each colored bead is going to represent a different amino acid, and we have 4 amino acids that we're including in our model for the purposes of this model. But there are more than 4, but there are 4 core amino acids that make our protein, and these 4 are going to be glycine, proline, hydroxyproline, and arginine. So first we're going to talk about glycine. Glycine is a simple amino acid that has a hydrogen atom in its side chain, and it's super small and flexible. So we have proline. Proline is a cyclic amino acid, and it has a side chain that bonds to nitrogen. And this structure is going to form, this amino acid is going to help form the kinks and turns in the triple helix structure. And then we have hydroxyproline. So this is a derivative of proline, the one that we just talked about, but instead it has an OH group added to a carbon 4 on the proline ring. This is essential in stabilizing the triple helix and making sure that it's strong. And then lastly, we're going to talk about arginine. Arginine is a basic amino acid with a guanidino group. It's positively charged, and this is important in ionic interactions. And this guanidino group also makes it highly hydrophilic. So now I'm going to talk a little bit about the mutated amino acid sequence. The only difference is, so in the mutation, as I said, we have cysteine substituting in for arginine. So I'm going to talk about cysteine being one of the, being the amino acid that changes in the mutation. So cysteine is a sulfur-containing polar amino acid with a thiol group in its side chain, and it's chemically reactive and forms these disulfide bonds. Now, these disulfide bonds are what's going to cause issues, because we had, if you think about arginine, arginine was a positively charged ion that was important in interactions between ionic compounds, and now we're removing this and exchanging it for something that can form disulfide bonds, which is not exactly the same thing. Disulfide bonds are not in the normal amino acid sequence, and this is going to distort the normal structure of the collagen triple helix, and this can also cause the misfolding of collagen molecule. So this can also prevent the proper assembly and weaken the properties of bone and skin, since collagen is such an important factor in our bone formation and bone structure in our skin. So this can also prevent the proper alignment of the collagen chains in the triple helix, and create a structurally unstable collagen molecule, which, as you can imagine, can cause all sorts of problems and also be detrimental to our bones and skin and everything that we have with collagen in it. So now with that overview of our protein molecule, so as I was saying, now with this overview of our protein, I'm now going to talk and show some examples of models and talk a little bit more about the structure of our protein and how we see this in the models that we have created. So first I'm going to talk a little bit about primary structure. In primary structure, this is our model for primary structure. In primary structure, you can see our amino acids joined by our polypeptide chain. One limitation for this model is I don't have a way to see the mutation on this, so you would just know that one of the, arginine, the amino acid arginine would be substituted for cysteine. So now we have our secondary model. It's a little different than the first. Here we can see our 3D structure that's formed by the folding of our polypeptide chain. This is an alpha helix, and it's stabilized by hydrogen bonds between the backbones of atoms. So here we have our tertiary structure. This is going to be our overall 3D shape. So here we have our triple helix, and we can see that this is 3 alpha helix chains connected together by hydrogen bonds. Our yellow pipe cleaners here represent our hydrogen bonds that hold all of our alpha chains together, and then we still have our amino acids represented by the beads on our alpha helix chains. Now, when we look at this model and think about mutations, one limitation to our tertiary model is that you can't see exactly what happens when a mutation like the cysteine arginine substitution occurs, but we would just know that when the substitution occurs, these bonds are going to be looser and this structure is going to either fall apart or be weaker, and it's not going to be as tightly bound as it normally would be. Lastly, I'm going to briefly talk about quaternary structure. So in quaternary structure, we still have our the same structure that we have from tertiary with our triple helix, but the only difference is in quaternary structure, other molecules from the environment are able to interact with our triple helix. So this is the assembly of multiple polypeptide chains, and it can build a functional protein complex. So this could either be multiple triple helixes joining together, or say just a random molecule decides to bond. That could also mean that it's able to interact with that, and then you'd have structures all hooked together. And this quaternary structure is stabilized by the same interactions as tertiary, so there's nothing new or added on that is occurring in quaternary structure. Lastly, I'm going to talk about one more limitation that exists within the use of our models. So as I talked about the specific properties that have to do with the amino acids that are a part of our protein, these chemical properties that are specific to the amino acids are not able to be represented fully by our models or in our models. Obviously, in our models, all of our beads are the same size and shape, and you can't see how they affect the bonds and properties of the triple helix. So these properties just aren't able to be, you're not able to see how they truly affect the models in our examples. Thank you so much for watching.